Welcome back, my dear students. Today we are going to discuss fertilization in angiosperms. So, fertilization it is simply defined as the fusion of male and female gametes to form a diploid zygote. Okay. Now the question is, why are male and female gametes produced in angiosperms? The male gametes are produced inside the pollen grain or the male gametophyte, while as the female gamete, or you can say egg, it is produced inside the female gametophyte or embryo sac. And in addition to egg cell, the female gametophyte or embryo sac, it also has the polar nuclei which are situated in the middle, right in the middle of the embryo sac. And besides these two structures, I mean egg cell and polar nuclei, it also has synergids, it also has antipodal cells. We have already talked about these details in the previous classes. Now, in angiosperms, the male gametes, they are transferred from pollen grain to the deep-seated egg in female gametophyte. जो egg होता है वो female gametophyte में होता है और female gametophyte itself is lodged inside the ovule. So in angiosperms, the male gametes, they are transferred from pollen grain or male gametophyte to the deep-seated egg in female gametophyte through pollen tube, which is developed by the pollen grain. Okay, and this transfer of sperm or male gametes from pollen grain or male gametophyte to egg through pollen tube, this is known as sphenogamy. So sphenogamy is defined as the transfer of pollen male gametes from male gametophyte to the egg cell situated in the embryo sac through pollen tube. Sphenogamy was first of all reported by Strasburger. It was discovered by Strasburger for the first time. And the pollen tube was first discovered by G.B. Amici in Portulaca. Portulaca is a plant in which for the first time pollen tube was discovered by G.B. Amici. Now there are different steps which occur prior to fertilization in angiosperms. We will try to discuss these steps one by one. The first step that we are going to discuss is the germination of pollen grain on stigma. Germination of pollen grain on stigma. You know that a large number of pollen grains, they can land, they can fall on the stigma. बहुत सारे pollen grains fall कर सकते हैं, बहुत सारे pollen grains गिर सकते हैं stigma पे. And when these pollen grains, they drop on the surface of stigma, abruptly there occurs an interaction between the pollen grain and the tissues of the pistil and the tissues of the stigma. This type of interaction, this is known as pollen pistil interaction. And the pollen pistil interaction, it determines, it tells you whether the pollen grain will germinate or its germination will be inhibited. You already know that some pollen grains are compatible and some are incompatible. This compatibility and incompatibility, it is determined by the pollen pistil interaction. The pollen and the pistil, they recognize whether the germination will be promoted or it will be inhibited. So only compatible pollen grains are able to germinate and grow. They first of all absorb the nourishment and swell up. The nourishment which is present on the surface of stigma, it is absorbed by the pollen grain and the pollen grain swells up. The swollen tube cell which is present inside the pollen grain, it grows out of the pollen grain through one of the germ pods. अगर हम बात करेंगे pollen grain structure की, pollen grain कुछ ऐसा होता है, इसमें दो cells होती है, एक को हम बोलते हैं tube cell और दूसरे को हम बोलते हैं generative cell. Okay, and this pollen grain, its wall is composed of two layers, one is known as axene and second one is known as intine. Intine अंदर की ओर होता है और axene बाहर की ओर होता है. This is the axene. At some places you will find that there is no axene, only intine is present. These regions constitute the germ pores. These regions constitute the germ pores. At these regions, at some of these regions, the intine, it protrudes out in the form of a long tube-like structure which is known as pollen tube. Okay, so obviously the pollen tube, it is having only one layer, that is intine. There is no axene in the pollen tube. Now both these cells, I mean tube cell and the generative cell, they move into the pollen tube. जब ये pollen tube में move करती है, okay, this generative cell, it divides and forms two male gametes. It forms two male gametes. This is one and this is second. These two male gametes, they are the result of the division in the generative cell. Now you have three cells in the pollen tube. Okay. The generative cell divides and forms two male gametes if it has not divided already. मैं आपको बताता चलूँ कुछ species में क्या होता है, before pollination generative cell divides and forms two male gametes. However, in case of some other species, the generative cell divides only after pollination. This is the only difference. Generative cell divide होती है, कुछ species में देखा गया है it divides when pollination has not taken place, but in other cases it divides only after pollination occurs. Okay, so this pollen tube, then it starts migrating through the stylar tissues here and reaches the ovule and enters the female gametophyte or embryo sac. Okay, now we will discuss how the pollen tube enters the ovule and ultimately the female gametophyte. The entry of pollen tube in the ovule, it is of three types. It differs in different species. The first one is porogamy. Porogamy simply means the pollen tube enters through micropyle, and this is the most common type of pollen tube entry observed in angiosperms. Example is that of lily. You can find this type of pollen tube entrance in lily. The second one is mesogamy. In this type of pollen tube entrance, the pollen tube enters through the funiculus or through the integuments. Here you can see the pollen tube is entering through the integuments. Example is Cucurbita. And the final one is chalazogamy. In this type of pollen tube entrance, the pollen tube enters the ovule, the embryo sac, through the chalazal end of the ovule. Here, example is Casuarinia and Juglans. Now you can see in the images the three types of pollen tube entrance. This is porogamy through the micropyle. This is chalazogamy. Sorry, this is mesogamy through the integuments or the funiculus, and this is chalazogamy through the chalaza. So with this we have completed how pollen tube germinates, okay, then how it enters the embryo sac. In the next part we will discuss double fertilization. Okay, we will discuss double fertilization and the significance of double fertilization in case of angiosperms.